As single-cell organisms, bacteria are exposed to the environment all the time, and one big challenge for them is dealing with viral infections. During the infection, viruses attach to the bacterium and inject the DNA into the cell. And if the bacterium's well-being is at stake, a rapid and accurate response is needed for self-protection. But the question is, how can bacteria distinguish foreign DNA from its own DNA? Of course, our smart bacteria have several ways to solve this problem. But here I want to introduce one important defense system against foreign DNAs in bacteria, the restriction-modification system. It can identify DNA based on both sequence and methylation patterns. And two key players in this system are a methylation protein for methyl group transfer onto DNA bases, and a restriction protein for DNA double-strand cleavage. Here is a typical modification protein, the E-core124. It seems to be solid in its structure, but if we rotate it, we can see some holes on its surface, and these holes provide access to DNA molecules. Actually, the E-core124 is not a single protein, but is rather made of 4 subunits, and the cleft formed by these subunits is large enough to accommodate a DNA molecule. Importantly, the binding of the modification protein to DNA is not random but sequence-specific. Once the DNA is recognized by the protein, the DNA bases ready for methylation flip out and enter the protein's catalytic pockets. Thus, the methyl groups will only be added to specific bases in the DNA that are part of the enzyme's recognition sequence. Once DNA modification is completed, the DNA will have a certain methylation pattern that protects it from cleavage by the restriction enzyme that I will introduce next. Restriction enzymes are proteins that bind and cut DNA, and that binding is also sequence-specific. This means that if the DNA sequence is not what the restriction enzyme can recognize and bind, it will not be cut. Importantly, if the DNA target sequence is methylated by the modification protein described earlier, it is protected from cleavage. In contrast, foreign DNA, such as that of an invading virus or plasmid, contains no methylation or maybe carries a wrong methylation pattern. That DNA will be recognized by bacteria as non-self, cleaved by the restriction enzyme, and degraded by nucleases. Now you know how a bacterium strikes back and successfully protects itself. Actually, bacteria can do much more than you think. It's worth mentioning that bacterial restriction modification system exists in many different flavors. In fact, one bacterium may deploy multiple RM systems in parallel. They are incorporated with one another, together making for a strong barrier against foreign DNA, and along the way play many other functions, like controlling the rate of evolution of the organism, and even increasing the relative fitness of bacterial hosts in the population. However, our knowledge of RM system is still limited. We still don't know if there is a specific rule for the establishment of distinct methylation patterns, and the identification of new RM genes remains a difficult task in scientific research. From where we are standing, there is a long journey for scientists to travel to understand this amazing defense system bacteria use to survive in a sometimes hostile world.